Assalamu alaikum. Dear students, I'm here Hafiz Muhammad Ammar, co-teacher for course Microbiology, code BOTN 4128. Today, lecture number 22, we will discuss phases of the growth, which will include lag phase, log phase, stationary phase, and a death or decline phase. Bacteria are prokaryotic organisms that most commonly replicated by asexual process of binary fission. We have already discussed in previous lectures. Binary fission and other cell division processes bring about an increase in the number of cells in a population. Population growth is studied by analyzing the growth curve of a microbial culture. When microorganisms are cultivated in liquid media, they usually are grown in a bath culture, batch culture, or a closed system, that is, they are incubated in a closed cultivated vessel with a single batch of medium. Because no fresh medium is provided during incubation, nutrient concentrations decline and concentrations of wastes increases. Consider a population of organisms introduced into a fresh, nutrient-rich media, a mixture of substances on or in which microorganisms grow. Such microorganisms display four major phases of growth: lag phase, the log phase, also called logarithmic phase, and the stationary phase, and the last one, the decline phase, or you can also say death phase. Bacterial growth curve, as you can see in the picture. The bacterial growth curve represents the number of live cells in a bacterial population over a period of time. There are four distinct phases of the growth curve. Lag, also called exponential phase, log phase, the stationary phase, and the death phase. The initial phase is the lag phase, where bacteria are metabolically active but not dividing into two. The exponential or log phase is a time of exponential growth. In the stationary phase, growth reaches a plateau as the number of dying cells equals the number of dividing cells. While in the death phase, which is characterized by an exponential decrease in the number of living cells. We will discuss one by one all these phases of the growth. The lag phase. The initial phase is characterized by cellular activity but not growth. When microorganisms are introduced into a fresh culture media, usually no immediate increase in cell number occurs. So this period is called the lag phase period. Although cell division does not take place right away and there is no net increase in mass, the cell is synthesizing new components. A lag phase prior to the start of cell division can be necessary for a variety of reasons. The cells may be old and depleted of ATPs, essential cofactors, and ribosomes. These must be synthesized before growth can begin. The medium may be different from one the microorganism was growing in previously. Here, new organisms would be needed to use different nutrients. Possibly the microorganisms have been injured and require time to recover. Whatever the causes, eventually the cells retool, replicate their DNA, begin to increase in mass, and finally divide. The lag phase varies considerably in the length with the condition of the microorganisms and the nature of the medium. This phase may be quite long if the inoculum is from an old culture or one that has been refrigerated. Inoculation of a culture into a chemically different media also results in a longer lag phase. On the other hand, when a young, vigorously growing exponential phase culture is transferred to a fresh media of the same composition, the lag phase will be short or even maybe absent. After the lag phase, bacterial cells enter the exponential or a log phase. This is the time when the cells are dividing by binary fission and doubling in numbers after each generation time. Metabolic activity is high as DNA, RNA, cell wall components, and other substances necessary for growth are generated for division. In this growth phase, that antibiotics and disinfectants are most effective as these substances typically target bacterial cells or the protein synthesis process of DNA transcription and RNA translation. Once organisms have adapted to a media, population growth occurs at an exponential or logarithmic rate. When the scale of vertical axis is logarithmic, growth in this log phase appears on a graph as a straight diagonal line, which represents the size of bacterial population. 1 base 10 logarithmic scale, each successive unit represents a tenfold increase in the number of organisms because each individual divides at a slightly different moment. The growth curve rises smoothly rather than in discrete jumps. The population is most uniform in terms of chemical and physiological properties during this phase. Therefore, exponential phase cultures are usually used in biochemical and physiological studies. During the log phase, the organisms divide at their most rapid rate, a regular genetically determined interval which is called the generation time. The population of organisms doubles in each generation time. For example, a culture containing 1000 organisms per milliliter with a generation time of 20 minutes would contain 2000 organisms per milliliter after 20 minutes, 4000 organisms after 40 minutes, and 8000 after 1 hour, and 64,000 after 2 hours. And if continue for 3 hours, it will be a 512,000. Such growth is said to be exponential or logarithmic. The generation time for most of the bacteria is between 20 minutes and 20 hours, and it is typically less than 1 hour. Some bacteria, such as those that cause tuberculosis and leprosy, have much longer generation times. Some individual cells take slightly longer than others to go from the lag phase to the log phase, and they do not all divide precisely together. If they divided together and the generation time was exactly 20 minutes, the number of cells in a culture would increase in a stair-step pattern, exactly doubling every 20 minutes. In a hypothetical situation which is called synchronous growth. In an actual culture, each cell divides sometimes during the 20-minute generation time with about 1 by 20 of the cell dividing each minute. This is a natural situation which is called non-synchronous growth. Non-synchronous growth appears as a smooth line, not as steps on a graph. Organisms in a tube of culture media can maintain logarithmic growth for only a limited time. As the number of organisms increases, nutrients are used up, metabolic wastes are accumulated, living space may become limited, and aerobes may suffer from oxygen depletion. Generally, the limiting factor for logarithmic growth seems to be the rate at which energy can be produced in the form of ATP. As the availability of nutrients decreases, the cells become less able to generate ATP and their growth rate decreases. The decrease in growth rate is shown in a gradual leveling off of the growth curve. You can see in the graph. A growth curve for an exponentially increasing population plotted for a synchronously dividing population shown in red line, and for a non-synchronously dividing population which is shown as green line. The blue spheres represents the number of bacteria present in each generation after beginning with a single cell. Exponential growth is a balanced growth. That is, all cellular constituents are manufactured at constant rates relatively to each other. If nutrient levels or other environmental conditions change, unbalanced growth results. This is growth during which the rates of synthesis of cell components vary relative to one another until a new balanced rate is reached. Unbalanced growth is readily observed in two types of experiments. Shift up, where a culture is transferred from a nutritionally poor medium to a richer one, and shift down, where a culture is transferred from a rich medium to a poor one. In a shift up experiment, there is a lag while the cells first construct new ribosomes to enhance their capacity for protein synthesis. This is followed by increase in protein and DNA synthesis. Finally, the expected rise in reproductive rate takes place. In shift down experiment, there is a lag in growth because cells need time to make the enzymes required for the biosynthesis of unavailable nutrients. Consequently, cell division and DNA replication continue after the shift down, but net protein and RNA synthesis slow. The cells become smaller and recognize themselves metabolically until they are able to grow again. Then balanced growth is resumed and the culture enters the exponential phase. These shift up and shift down experiments demonstrate that microbial growth is under precise, coordinated control and responds quickly to changes in environmental conditions. When microbial growth is limited by the low concentration of a required nutrient, the final net growth or yield of cells increases with the initial amount of the limiting nutrient presents. This is the basis of microbiological assays for vitamins and other growth factors. The rate of growth also increases with nutrient concentration, but in a hyperbolic manner, much like that seen with the many enzymes. The shape of the curve seems to reflect the rate of nutrient uptake by microbial transport proteins. At sufficiently high nutrient level, the transport systems are saturated and the growth rate does not rise further with increasing nutrient concentrations. Leveling off of growth is followed by the stationary phase. Unless fresh media is added, organisms are transferred to fresh media. Logarithmic growth can be maintained by a device much like thermostat, and this device is called chemostat. You can see in figure, which has a growth chamber and a reservoir from which fresh media is continuously added to the growth chamber as old media is drawn. Alternatively, organisms from a culture in the stationary phase can be transferred to a fresh medium. After a brief lag phase, such organisms quickly re-enter the log phase of the growth. Because this is a closed system, eventually population growth ceases and the growth curve becomes horizontal. This stationary phase usually is attained by bacteria at a population level of around 10 power is 9 cells per mil. Other organisms normally do not reach such high population densities. Protists cultures often have maximum concentrations of about 10 power is 6 cells per ml. Of course, final population size depends on nutrient availability and other factors as well as the type of microorganism being cultured. In the stationary phase, the total number of viable microorganisms remains constant. This may result from a balance between cell division and cell death, or the population may simply cease to divide but remain metabolically active. Microbial populations enter the stationary phase for several reasons. One obvious factor is nutrient limitation. If an essential nutrient is severely depleted, population growth will slow. Aerobic organisms often are limited by oxygen availability. Oxygen is not very soluble and may be depleted so quickly that only surface of a culture will have an oxygen concentration adequate for growth. The cells beneath the surface will not be able to grow unless the culture is shaken or aerated in another way. Population growth also may cease due to the accumulation of toxic waste products. This factor seems to limit the growth of many aerobic cultures. For example, streptococci can produce so much lactic acid and other organic acids from sugar fermentation that their medium becomes acidic and growth is inhibited. Streptococcal cultures can enter the stationary phase due to the depletion of their sugar supply. Finally, there is some evidence that growth may cease when a critical population level is reached. Thus, entrance into stationary phase may result from several factors operating in concert. As we have seen, bacteria in a batch culture may enter stationary phase in response to starvation. This may be occur in nature because many environments have low nutrients. Prokaryotes have evolved a number of strategies to survive starvation. Many do not respond with obvious morphological changes such as endospore formation, but only decrease somewhat in overall size, often accompanied by protoplast shrinkage and nucleoid condensation. The more important changes are in gene expression and physiology. Starving bacteria frequently produce a variety of starvation proteins, which make the cell much more resistant to damage in a variety of ways. They increase peptidoglycan cross-linking and cell wall strengths. The DNA binding proteins from starved cells protects the DNA. Chaperone protein prevent protein denaturation and renature damaged proteins. As a result of these and many other mechanisms, the starved cells become harder to kill and more resistant to starvation itself. Damaging temperature changes, oxidative and osmotic damage, and the toxic chemicals such as chlorine. These changes are so effective that some bacteria can survive starvation for years. This is even evidence that Salmonella enterica, Servor, Taphimurium, and some other bacterial pathogens become more virulent when starved. Clearly, these considerations are of great practical importance in medical and industrial microbiology. For many years, the decline in viable cells following the stationary cells was described simply as the death phase. It was assumed that detriment environmental changes like nutrient deprivation and the buildup of toxic waste cause the irreparable harm resulting in loss of viability. When bacterial cells were transferred to fresh media, no cellular growth was observed because loss of viability was often not accompanied by a loss in total number cell. It was assumed that cells died but did not lyse. This view is currently under debate. There are two alternative hypotheses you can see in the picture. Some microbiologists believe starving cells that show an exponential decline in density have not irreversibly lost their ability to reproduce. Rather, they suggest the microbes are temporarily unable to grow, at least under the lab conditions. This phenomena, in which cells are called viable but non-culturable, is thought to be the result of genetic response triggered in starvation, stationary phase cells. Just as some bacteria form spores as a survival mechanism, it is argued that others are able to become dormant without changes in morphology. Once the appropriate conditions are available, for example, change in temperature or passage through an animal, these microbes resume growth. These microorganisms could pose a public health threat as many assays that test for food and drinking water safety are culture based. The second alternative to simple death phase is programmed cell death. In contrast to the viable but not culturable cells hypothesis, cells are genetically programmed to survive. Programmed cell death predicts that a fraction of the microbial population is genetically programmed to commit suicide. In this case, non-culturable cells are dead as opposed to non-culturable, and the nutrients that they leak enable the eventual growth of those cells in the population that did not initiate suicide. The dying cells are the uh, altruistic, that is to say, they sacrifice themselves for the benefit of the larger population. As conditions in the medium become less and less supportive of cell division, many cells lose their ability to divide. Thus, the cells die. In this decline phase or death phase, the number of live cells decreases at a logarithmic rate, as indicated by the straight downward sloping diagonal line in earlier picture of the growth phases. During the decline phase, many cells undergo involution, that is, they assume a variety of unusual shapes, which makes them difficult to identify. In cultures of spore forming organisms, more spores than vegetative cells survive. As dying cells lyse or break open, they spill their contents into the environment, make these nutrients available to other bacteria. This helps spore producing bacteria to survive long enough for spore production. Spores are able to survive the harsh conditions of the death phase and becoming growing bacteria when placed in an environment that supports the life. The duration of this phase is as highly variable as the duration of logarithmic growth phase. Long-term growth experiments reveal that an exponential decline in viability is sometimes replaced by a gradual decline in the number of culturable cells. This decline can last months to years. During this time, the bacterial population continually evolves so that actively reproducing cells are those best able to use the nutrients released by their dying brethren and best able to tolerate the accumulated toxins. This dynamic process is marked by successive waves of genetically distinct variants. Thus, natural selection can be witnessed within a single cultural vessel. Figure shows that instead of distinct death phase, successive waves of genetically distinct subpopulation of microbes better able to use the released nutrients and accumulated toxins survive. Each successive solid or dashed curve represents the growth of a new subpopulation. In this figure, during the development of bacterial colonies, the amount of movement depends on the growth phase. In first row, a droplet of bacterial solution, E. coli, on an agar plate grows into colonies. The growth movement accelerates from the lag phase to the exponential phase with the duration of hours. You can see in 5 hours time the exponential phase achieved. Then slows down and stops reaching the stationary phase. You can see in the 18 hours the stationary phase reached. In B line, you can see the microscopic pictures of the bacterial samples. The bacterial area expands and fully covers the surface around the exponential phase and forms a denser colonies until the stationary phase reaches. The arrows in the picture indicate that the amount of dynamics at a specific time depends on its phase being most vigorous at time 5 hours. In C line, you can see picture of the agar plate with bacteria for each time. The colonies are visible only after the end of the exponential phase at 7 hours. These are the reference links, books for where the lecture was prepared. You can also study these lectures, these reference materials. Thank you very much.